Hi everyone, welcome to my channel Pure Biology and I am Tapushi, and today I come with the continuing video of mitochondria. This is the second part of the mitochondria, okay? And the first part of the mitochondria I have already uploaded in my channel, okay? So you just check out that one, okay? And this is the continuing part of the mitochondria, or you can say the second part of the mitochondria, clear? And before going to this topic, I want to say that thank you to all of you who have already subscribed my channel. And if this channel is helpful to you, or then you obviously subscribe and share this channel with your friends. And after subscription, you just hit the bell icon for getting the notifications of any video that will upload in that channel, okay? So let's continue the mitochondria. In the first part, we have learned that the mitochondria, generally these are developed from the endosymbiotic or endosymbiotic part, not symbionts, okay, or symbiotic cell. And this is the actually here I just written here just some evidences why this is called the mitochondria generally developed from the endosymbionts. Clear? So the evidences are, okay, and the evidences are that mitochondria are the self-replicating bodies like bacteria and divide in a manner resembling the binary fission in bacteria. Okay, so one thing is that these are the self-replicating organelle. That means they have its own DNA, okay, or their own DNA present within that organelle, and that is not similar to the DNA that present in the nucleus. And obviously these are produced by themselves in the process of binary fission like bacteria. And second point is that mitochondria are surrounded by the two membranous structure. One is the outer membrane and another is the inner membranes. And here most of these membranes very similar in composition to the bacteria. That means the inner membrane and the outer membrane already that is surrounded the mitochondrial part or mitochondrial structure, the composition of that membrane generally is similar to the composition of the bacteria. Clear? And third point is that mitochondrial DNA is similar to bacterial DNA. Okay, this is very similar because bacterial DNA size is very small, just like mitochondrial DNA, and shape is circular, and same type of DNA is seen in the mitochondria. Okay, and next point is that mitochondria are approximately the same size as bacteria. I am saying that the shape and size both are similar to the bacteria. Okay. And protein synthesis is inhibited by the antibiotics in case of mitochondrial DNA or mitochondrial protein synthesis, that is inhibited by some antibiotics. What antibiotics are there? Tetracycline and erythromycin. And these antibiotics are responsible for the inhibition of the protein synthesis of bacteria. Clear? And the next point, no, this is the last point. So actually, if these evidences are just the structure of these evidences actually said that the structure of mitochondria and the properties of the mitochondria that are very similar to the bacterial cell. Okay, so that's why these are produced. Generally scientists that according to the scientist opinion, these are produced from the bacteria. Okay, which bacteria? Generally photosynthetic bacteria, and during the evolution, okay, or they evolved from the bacterial cell during evolution. Clear? So that's why this endosymbiotic theory is generally is the actual opinion of the scientist that the mitochondria are produced or evolved from the endosymbionts. Okay. Next part is actually related to the hydrogenosome and mitosome. Some protist, okay, some protist and sometimes some unicellular organisms, they haven't mitochondria, and instead of mitochondria they have hydrogenosome and sometimes mitosome. So what are actually hydrogenosome and mitosome? Hydrogenosome is actually just like mitochondria and they function like mitochondria, and where it is seen, example is trichomonas species. Okay, so here the hydrogen and ATP both are synthesized in this hydrogenosome. Okay, and here we know that in mitochondria during ATP production CO2 is produced, but here hydrogen is produced in molecular form. Okay, hydrogen is produced molecular form. And these are the sites of the pyruvate fermentation. The pyruvate fermentation also is possible in here. Okay, and ATP production also is produced in this pyruvate fermentation, and here the process is occurring through the substrate level phosphorylation. Substrate level phosphorylation. So what is happening here? That pyruvate fermentation procedure is seen here, and obviously ATP is produced, and the process occurring through the phosphorylation process. Okay, phosphorylation process. And here what is happening, that pyruvate that is broken down to acetate CO2 and H2. Oh, okay. I just said that CO2 is not produced here, but not that is true. CO2 is also produced here. With CO2, hydrogen is produced, but that is not produced in the actual mitochondria. Clear? That means CO2 is produced here, obviously with hydrogen, but hydrogen is not produced in general aerobic respiration process in mitochondria. Clear? In contrast to mitochondria, this is saying here that in contrast to mitochondria, this produce the hydrogen through the fermentation and lack cytochromes and DNA. One main difference is that the hydrogenosome that never contains DNA, but mitochondria, they have obviously their own DNA. Okay, and hydrogen is produced in the hydrogenosome, but in mitochondria the hydrogen is not produced. Next one is the mitosome, and where it is seen, generally mitosome are found in entamoeba. Okay, there is not found mitochondria. Actually mitochondria is generally found highly organized cell, obviously highly organized cell. These are obviously the lower unicellular cells, and proteins, these are obviously lower cells. So that's why sometimes the mitochondria are absent here. And here this mitosome is sometimes called the reduced mitochondria or cryptic mitochondria. Clear? This is the double membranous structure, obviously double membranous structure. Okay, so these are the actual evidences: hydrogenosome, mitosome. Next part is that targeting of mitochondrial proteins. Targeting of mitochondrial proteins. Okay, so this mitochondrial proteins import, this requires generally proteins remain in where, if that is present in matrix, inner membrane or outer membrane. So this protein imports possible through the receptors and translocons. Clear? receptors and translocons. With the help of these, the import of mitochondrial protein is possible. Clear? And this import is initiated by the binding of a mitochondrial targeting sequence to an import receptor in the outer membrane. Okay? So, targeting sequence and receptor both bind to each other, then transport is possible. And two distinct translocation complex present in the outer and the inner membrane. One is called the TOM, another is called the TIM. TOM is the translocase of outer membrane and TIM is the translocase of inner membrane. So, with the help of these two translocases, the actual transport or import, not transport, import you can say, import of the proteins is possible. So, what is actual TOM or TOM and TIM or TIM is what? So, we will learn about that in the next video. Clear?